Good morning, everyone. Now it's my great honor to introduce Mr. Pritam Kumar Panda, sir. He is assistant with doctoral in the Department of Physics and Astronomy, Uppsala University, Sweden, under the supervision of Professor Rajiv Ahuja and Professor Yogendra Kumar Mishra from Mads Clausen Institute, Nano SYD, University of Southern Denmark. He has worked as a junior research fellow in Biology Lab, KIIT University, India. His expertise includes computational biology, biophysics, 2D material modeling of electron transport in nanoscale device, and computational modeling of bio nanoparticles. He has published more than 57 papers, research papers. So I would like to invite Pritam Kumar Panda, sir, for his talk. Please welcome, sir.
Thank you very much for the introduction. It was a nice and pleasure to be here. I'm going to share my presentation draw. So, can you see my screen?
Yes, Dr. Panda. We are able to.
Yeah.
Why?
Yeah, okay. So, good morning, all of you. It's very early morning here. I think in India it's 11:15 over there. So, good morning, everyone. I am Pritam. I'm a researcher in Uppsala University, and I am from the materials theory group here in Uppsala University, working under the supervision of Professor Rajiv and Professor Yogendra Mishra from Southern University of Denmark. So today's topic will be regarding the genome to genome. This is the term that I coined during my PhD. And I gave a catchy moniker to do some translational research interfacing molecules with nanomaterials. So this will be a general talk. I mean, I'm not going to deep dive into the results or something. I just give you an overview. What are my works that has been done over here, and why this term I have given like genome to genome, and how I have done some translational research on interfacing molecules with different nanomaterials. It ranges from zero-dimensional materials to three-dimensional materials as well. So, and the catchy moniker that you see, that genome to genome, it's a term, as I mentioned, that I coined by me, and by bridging the concept of biophysics and quantum physics, which abbreviates to genome, genetic organization of the multicellular organism and their enzymatic reaction, to quantum nanostructured materials for energy scavenging applications. And it's a combinatorial approach using computational methodology in biophysics and in material science, where I study the interface of the molecules interacting with different type of nanomaterials. So today's topic that I'm going to cover based on three main aspects. One is electronic transport at the nanoscale, where I do biosensing of DNA, amino acids, and different gas sensing. Number two is the contact electrification with these nanogenerators. And the third topic would be nanomaterials with different complex, architectures, like as you see on the right-hand side, the zinc oxide tetrapods, So these are the three main areas where I am working on, and these are some of the topics that I'm going to, cover today. So different types of applications are there. You see the first topic, that is biosensing or electronic transport at the nanoscale. I think Dr. Burma is one of the most searched, tutorial. If you want to learn electronic transport, then you go to Dr. Burma's tutorial on YouTube, and then next comes to me, because I do also some tutorials inspired by Dr. Burma. So Regarding the first application, that is solid state sequencing, as we say, this is applied for personalized medicine, environmental sensors, and different water purifiers as well. The materials that I have taken, 2D materials, they are also widely used for different water purification systems as well. And the second topic, that is contact electrification, the term that is coined by Professor Zhonglin Wang. And I have been fortunate enough to work with him. He's one of the great scientists and the scientist behind the term turboelectricity or contact electrification. This can be utilized for sensor for smart home lighting, smart wearable devices, drug delivery, and different power transformers. The third thing, which I work with Professor Yogendra from Southern University of Denmark, is based on nanomaterials with complex architecture that is based on 3D materials, which is called zinc oxide tetrapods. And that was a very good antiviral agent. And whatever the research that I'm doing, those are targeting to the sustainable, one of the key goals of sustainable aspects, that is life below water. So different types of computational methodology I use, because this is a combinatorial technology. So for the density functional theory, I use VASP, and for electronic transport, that was inspired by Dr. Verma's YouTube, is TransIESTA. And I use some classical dynamics, because dealing with billions of atoms and millions of atoms is not possible in DFT. That's why I use classical dynamics such as molecular dynamics, ab initio molecular dynamics with VASP, dealing with few atoms, to classical molecular dynamics using GROMACS and LAMMPS. And they use different types of parameters in order to get some insights into their properties. Okay. So I'll talk about electronic transport at the nanoscale. This is quite a wide topic, and I think the workshop is based on electronic transport calculation using Siesta, and in general, the Siesta use. So I have used DNA biosensing techniques, protein biosensing techniques, those are amino acids, and gas molecules. There are different toxic molecules in the environment. So those gas molecules has been sensed by using different types of 2D materials. So the materials that I have used is gallium sulfide, and carbon nitride is one of the analog of graphene, which can be used for water purification system. And bismuthine, particularly bismuthine, has been used for sensing the different gas molecules. As you can see on the right hand side, this is the nanomolecular electronic setup. And this has been done by using gold nanoparticles, and then bridged with the different types of molecular junctions like PPDT and TPDT. And it can be used for sensing different types of gas molecules. Okay. So the motivation came when I published a paper in Nature Medicine. I was working with the University of Freiburg, Germany. And what we have done is mostly next generation sequencing. The technology, the transport technology that you see over here, and whatever the DFT technologies that has been used, they are widely used in different universities, different companies, and they are used to treat different types of cancer patients. So in here, what you see in the Nature Medicine article is we have extensively used these machines, these Illumina machines. And I was baffled by the technology that has been used widely for next generation sequencing. And you can see that the technology that has been used in Illumina or any of the sequencing device is semiconductor-based technology. So that's why it allows me to work on different types of 2D materials, which has semiconducting behavior, and that allows me to do some transport calculation by using density functional theory. So in the right hand side, what you see is the typical setup of electrodes, 2 electrodes that we take of the same material or maybe from different material, and the scattering region where different molecules are being sensed. So this is a typical example of gallium sulfide monolayer with the DNA basis, which is adenine and thymine, guanine and cytosine. As you can see here, it has quite high range of sensing towards guanine and cytosine rather than adenine and thymine. I mean, this is the case for all the materials. I think whenever you do electron transport calculations with 2D materials, the sensing would be more towards guanine base pairs and cytosine base pairs rather than A and D. This is a typical result that you get. The next application is one of the peculiar application of different 2D materials. I have seen in 2020, University of Cambridge University, first time they proposed the amino acids that can be sensed in different 2D materials. So I have taken an analog of graphene, which is carbon nitride GC3N4 monolayer. The typical setup is almost similar. And I have sensed different types of amino acids. So there are 20 different types of amino acids and 2 different types of another amino acid. So in total, there are 22 different types of amino acids, including pyrrolysine and selenocysteine. So what we have observed over here, I mean, this technology has been used in water purifiers. So different water purifiers you have. So those purifiers, they use carbon nitride, the analog of graphene, that can sense the bacterial organisms or the bacterial proteins, and they can be used to filter them out. And by interacting with the analog of graphene, I mean, this graphene nitride has a good potential of an antibacterial agent. So the combination of density functional theory from here and a combination of molecular docking and molecular dynamics, where you molecular dock these 2D materials with different types of bacterial proteins, and that can affect the system, or that can kill the bacteria. So this is the technology. You can read my paper. I can send you the list of the papers which has been published based on the results that I'm showing. The next thing, this is a quite interesting setup. Here in Uppsala University, they have done some experimental analysis on that. And as you can see, they have mentioned me that, can you do some DFT calculations based on the experiment as a gas sensor? I said, yes, why not? Then I followed the tutorial from Dr. Verma and tried to establish the same. This is a nano MOAD, nanomolecular electrode junctions, where I use gold nanoparticles to construct the device with different types of gas molecules, sensed on the top by using a molecular junction called BPDT or TPDT. Okay, and this device can sense based on 2 different factors. One is chemisorption, another one is physisorption method. And this is a very good technology to sense the gas molecules. So this particular device is inclined towards NO2 sensor, nitrogen dioxide sensor, rather than NS3 sensor. Okay, I'm just going to show you one video by using Molden. That's one of the software for visualization, and from the transport calculation, how you can do some visualization on the basis of physisorption and chemisorption. So yeah, these are some of the results that you get from electronic transport. So one is current and voltage graph based on the different voltage parameters, and the transmission graphs, where you see at the Fermi level the rise of the peak, where different types of gas molecules can be sensed at the Fermi level. Okay, the high number of transmission peaks at the Fermi level, that means it has some types of resistance in the device. So this is one of the video that you can use by using Molden, and I think Dr. Verma is going to teach you about how the mechanism works and how you can utilize DFT analysis to visualize this kind of platform. So watch this video a little bit. You see the molecules and the junctions where the gas molecules have been adsorbed. One is chemisorbed and one is physisorbed. So you can see the conformation of the structure has been changed when it's chemijob, and during the physijob session, it's kind of stable. And this is based on the adsorption of chemijobson and physiojobson. As you can see, in case of TPTT, triphenyl diethyl and biphenyl diethyl, the triphenyl diethyl has a chemijobson on. Seen in the previous video, the conformation of the structure has been changed. Quite a lot, positive in binding energy, which is not good. Generally, what you get the binding energy is in negative areas, but this has a particular system for sensing nitrogen dioxide molecules. It kind of adsorbed on the surface of these type of molecules. And yeah, one of the application of solid state nanopore sequencing, nanopore sequencing is quite famous, and that has been devised by one Indian scientist here in Oxford. And you can see that these nanopores can be used also for sensing different types of DNA, amino acids, and even different gas molecules. But mostly it has been used for sensing DNA molecules. So this nanopore sequencing is also similar to whatever the technologies that has been widely used by Illumina. Okay, so what I've done is I've taken 2D materials, and this is the particular snapshot of a paper from Glavin et al. You can read this paper. This is a very nice paper describing how the p-block elements can be used to devise nanopores to sense different types of DNA molecules. So I have taken a series of the 2D materials and try to devise a nanopore based technology. You can find all the tutorials based on nanopore sequencing as well. It's there in my YouTube channel. So you can devise such kind of electrode setup or the scattering region setup, the transport setup, and then you can sense different types of DNA molecules. So in all the cases, as you can see, as I mentioned previously, it has quite a good sense for the guanine molecules, because guanine and cytosine almost they have triple head bonds, and they are slightly inclined to be interacting with different types of 2D materials. So the results in the up is based on DNA sequencing. I have used antimony, arsenine, bismuthine, germanium, MOS2, phosphorin, silicene, stannine, and you can use all different types of 2D materials to sense DNA molecules. And in terms of amino acid sequencings, it has been categorized into different categories like non-polar, polar amino acids. So every amino acid which has a functional R group, they are based on different types of functional groups, they are sensed towards different types of 2D materials. In terms of gas sensing, always you can find all the 2D materials, even if you do transport calculation or adsorption calculation as well, they are sensed towards nitrogen dioxide molecules. So yeah, very interesting results of what I found in my PhD, but you can also try to add on different types of 2D materials, and you can set up the devices for all these three different applications. Yeah. The next topic that you see on the left-hand side, the image, I think in the childhood, you might have done this. So when you rub a comb on your hair and try to identify some of pieces of paper, try to attach them. So you can see that's called static electricity. So this term has been developed by Professor Zhonglin Feng. And, he is one of the pioneers in triboelectric nanogenerators. This is one of the applications from the contact electrification application. So yeah, so I use this technology on different 2D materials. I have taken aluminum nitride, gallium nitride, and silicon to understand the mechanism of solid liquid interface. So in a solvent system, if you put the 2D materials, all these 2D materials, so what kind of effect that you are going to get? So I think in the solvent system, different types of, if you put these 2D materials for one of the applications in triboelectricity, so it has silicon has been quite widely used, and you can see that different molecules, they are present in the water, like NaCl, oxygen atoms, and different all different kinds of chlorine atom. And you can see that I have used a combination technology over here. On the upper hand side, what you see is density functional theory, where I did some total charge transfer calculation, beta charge calculations. And in the right hand side bottom corner, what you see is based on classical dynamics approach, where you can devise this kind of 2D material setup under a solvent environment and try to identify what type of adsorption principle they have for different 2D materials in solvent environment. So in this case, what you can see based on the lab's results, that chlorine is much more prominent on gallium nitride in comparison to other different types of 2D materials. So this is very interesting applications, and you can find the paper on solid liquid interface, which has been published in Nanoscale Advances. And yeah, this is one of the applications of contact electrification. So this is a basic application to understand the mechanism of solid liquid interface. But when you go for different types of applications, so you might have heard that gold nanoparticles, 2D materials, or even the clusters, they are widely used for different biological applications. So gold is one of the most powerful agent in drug delivery. So how it interacts, I mean, how can you devise such platform to understand different types of molecules, micromolecules inside your body, how they interact with the gold setup, or the gold nanoparticles. So this is a classic example where I've done molecular dynamics simulation using GROMACS to understand how these molecules, they get interacted on the surface of 2D materials. So this paper is not yet published, but I'm soon going to publish this paper. And you can see that these device setup can only be used when you use classical dynamics approach. And even in CSI also you can devise this, but it might take some time. But if you use LAMPS or if you use GROMACS for this kind of setup, then that would be really fantastic. Coming to another application of contact electrification. So you know that in power transformers where the electricity generation happens, nowadays they are using cellulose materials, and these cellulose materials are quite a good insulating agent. So so this project has been sponsored by Hitachi from here in Sweden, and they try to understand how the molecular interaction between different types of oil molecules used in power transformers, like naphthenic compounds, aromatic compounds, and paraffinic compounds. And they want to understand how these cellulose materials behave when exposed into different oil conditions. So as you can see in the left-hand side, that paraffinic compounds, they kind of form different types of clusters. So these oil molecules, they form different types of clusters. And this has been achieved by doing molecular dynamics simulation using GROMACS. And this setup you can use from JAM force field. If you know about classical dynamics, then you might have known about JAM force field. And this can be utilized to understand the solid-solid interaction interface. So this is quite a popular nuclear. This paper has not been published yet. This is under preparation, and I'm soon going to publish this paper so that you can know more details how I have done this. So this is not possible in density functional theory calculations. You have to use classical molecular dynamics. Okay, the next topic is very interesting to me. This has been supervised by Professor Yogendra Mishra from Southern University of Denmark. And this topic concerns about the 3D materials. As you can see that the animation that you see is typical structure of zinc oxide tetrapores. And it has a very complex architecture, like it has 4 arms and it attaches to a central point in zinc oxide tetrapores. So this is a very good antiviral agent. So one of the patents that we have done is this particular cream has been used for antiviral therapy. So whenever you get herpes virus, HSV1 is very common in world. So if you apply zinc uh, into that particular um, viral agents or infection, it can be cured within days. That's the power of zinc oxide. And you know that human body contains almost 30% of zinc. So zinc is very necessary. So it is a very good agent for antiviral agent. So what you have done in this work is we have taken zinc oxide tetrapods and coated with the different types of gold nanoparticles. And we just want to identify that whether this functionality of zinc oxide tetrapods remains the same, or the bare zinc oxide tetrapod has more activity. So what we have found is when we try to identify the interaction between different types of macromolecules from herpes simplex virus, and we can see that the zotin itself, the zinc oxide tetrapod itself, has high efficacy towards antivirals in comparison to the functionalization of the zinc oxide tetrapods. And during the pandemic, I also try to understand how the interaction of zinc oxide tetrapods with different types of viruses, and it is a very good agent for SARS-CoV-2 as well. So this can be a part of research, and this has been submitted in antiviral research. I think it will be published soon. So you may find, and this is a combinatorial technology of DFT. You have to construct zinc oxide tetrapod architecture by using density functional theory. Then you use, bioinformatics analysis like classical dynamics and classical molecular docking analysis to understand the interaction between different macromolecules. And if you use nanomaterials, you can use that as well. And this is a patent that we have filed, and I just want to show you some of the examples that whatever the things that we're doing in research in vivo. In vivo, it means we are using different organisms to understand how the nanomaterials interact. So this is a typical example of zebrafish embryos, which we used to study the interaction of zinc oxide tetrapods. So the arrow marks that you see, whenever it enters into different types of embryos, it stays on the surface. So if you put zinc oxide nanoparticles instead of zinc oxide tetrapods, then it might enter or invade different cellular layers, and it can cause reactive oxygen species and apoptosis. But zinc oxide tetrapods is a very nice toxic agent, I would say, and it gives less cytotoxicity to the cells instead of nanoparticles. So whatever the nanoparticles use, I'll show you one typical example of nanoparticles, that is silk nanoparticles, that has been used to study this kind of interactions. Yeah, and I just want to mention that whatever the results that you see on the surface of it, we just try to understand by using classical dynamics, using lamps, to study that whether these tetrapods, they remain attached when you cut off the arms in a solvent environment, and to see that whether these arms are getting attached again, or they have been detached in the same form in different water environment or the solvent environment. So yeah, this is a peculiar study. As you can see, when you cut off the 3 legs and try to simulate for more course of simulation or a longer time period of simulation, you can see that these arms are getting closer to the central point of zinc oxide tetrapods. It's a very nice and peculiar results that we found, and I think this is widely applied in practical scenarios as well. As I mentioned that, I just want to show you how these nanoparticles, they invade into the cells. And by using a combinatorial technology that is from DFT, as you can see from the left-hand side, these are silver nanoparticles. These are modeled by using density functional theory, and you can see total energy and the dynamics, how these nanoparticles, they behave in vacuum. But when it comes to the in vivo organisms, how these nanoparticles, they invade into the zebrafish embryos or the any particular organism, how they invade into the cells. And by using classical dynamics and technology, we can see that these are interacting and they induce a lot of reactive oxygen species and apoptosis, which might eventually kill the organism or kill the cells. So these nanoparticles are very, very toxic sometimes, but sometimes these nanoparticles, they do have a peculiar property without having any toxicity or cytotoxic to the cells. But in our case, it is quite, we have seen that this nanoparticle, they induce some kind of toxicity to the cells. And yeah, that's it. I mean, these are the three topics that I have covered. I mean, much more I have covered in my PhD, as you can see from my different papers. But I would like to thank Professor Y.K. Mishra, Jonglin Wong, Johan from Karolinska, Mrutyunjay, and Rajiv from Uppsala. And these are my colleagues who helped me to understand this concept, to frame the ideas, and to get some insights into biological perspective and from quantum physics perspective as well. Yeah, thank you very much. And you can watch my YouTube tutorials on Siesta, and you can combine both Dr. Verma's and mine YouTube tutorials to understand how the transport happens. I have a tutorial on the on the setup of different transport system, and you can find a detailed tutorial on that. So yeah, you can reach me out anytime, and thank you very much.
Now session is open for discussion. If anyone has any question, then you can ask.
Dr. Panda, it was really very attractive and wonderful lecture. Thanks for
Thank you.
Accepting the invitation from this organizing committee. And simple question related to that computational time. That compared to SISTA, the computational time of GROMACS is how you compare?
Very less. Very less.
So
yeah, and you can deal with the billions of atoms.
Okay, so it's easy to learn. I think I have the.
Yes, it's kind of easy to learn. Yeah, of course, of course, of course.
So we should suggest some others to do molecular dynamics summation using.
Oh yeah, yeah. Please. I mean, if you combine density functional theory with classical dynamics, either by using LAMMPS or maybe using GROMACS, that would be fantastic. That will give you a very nice overview of any of the results that you do, including 2D materials, including nanoparticles, or anything. You can do anything. So I have done recently published during the pandemic paper in Science, and I have used extensively molecular dynamics on that by using GROMACS.
Thank you.
Hello, Sir Panda. That was a wonderful and thought-provoking talk, as I would say. I have few questions, like if I can frame it out.
Yeah, please.
Yeah, like you talked about the K-desorption and P-desorption process, no, sir? Can we go back to that one? Like I saw the binding energy, like there was those two binding energy were nearly of the same order, no? So how did you distinguish that one as the P-desorption process and another one as the K-desorption?
Yeah, exactly. Good question. That this question has been asked by many experimentalists, and I would say this has baffled me as well when I try to implement this method to understand how the physisorption, the chemisorption works. So again, I have not shown one of the results because that one I have done by using molecular docking technology. Okay. So when you put the molecules on the top in Siesta or in BASP, when you take 2D materials and you put different types of molecules, those are based on distance-based methods. You randomly put, or else what you can do is you can do nudge elastic band calculation, NEB calculation.
Yeah, NEB calculation.
Okay. And you find out a common minima where the molecules is having a least energy or the lowest energy.
Yeah.
Right. And sometimes you see highest energy. So the highest energy point where in the 2D material, wherever you see, those are chemisorption state
Oh,
because they have a highest energy point. But
Yeah, yeah,
whenever
yeah.
you see a lowest energy point that has been,
Yeah.
quite favorable for 2D material,
Yeah.
that might be the case for the physics. I mean, you have to check. It depends on the
Yeah.
molecules and depends on how big is the molecule.
Mm.
So one of the key approach is NEB calculations or notch elastic band calculations.
Also, can we take into consideration the charge transfer mechanism also, like how much charge has been transferred from the molecules and
Yes,
all?
yes,
No? Like,
yes, yes,
yeah.
absolutely. So
You
in my
can-
in my case, in terms of nitrogen dioxide, when it was adsorbed on a specific molecule called TPDT, triphenyl diethyl,
Yeah.
so you can see that it forms a extra bond. So that extra bond is another way to understand the physisorption and chemisorption is work function. If your work function is high, that means it is.
Yeah.
If your work function is high, that means it is the very much energy is required to extract the bonding from that. So that's a typical example. If your work function is high, that means it is inclined towards chemisorption.
Okay. Yeah. Yeah. And also, sir, how did you design the solvent models, like those solvent models? And
The solvent
like
model.
how did you take into the consideration of those different atoms? Like how did you design the force field potential also? Like it's not all available in the literature and all. No, like how those two molecules interact and all.
Yeah,
Like
I
all
would
those
like
things are
to. I would like to tell you my secret. Why I've been using this. So you can use Charm GUI, CHARMM, Charm GUI. That's a very good platform to design materials for molecular dynamics simulation, which is based on 2D materials. You can design. So that's the platform. You go and the potentials have been defined there. So you don't have to do anything. You don't have to define any potential. You just directly go there and click on the 2D material that you are interested, and that's it. You can do that.
It will directly take into the consideration all those
Absolutely.
parameters.
And I have a tutorial on that also. So you can check my molecular dynamics tutorial.
I'll go
see.
through that one.
And
also,
if you follow my
please,
if you follow my GitHub channel, okay, there also there is a section called molecular dynamics, and you can see I have mentioned a protocol for nanomaterials also, like the cellulose materials that I've shown. I have a protocol for that, so you can also do that. And I'm also going to make some tutorials. I'm quite busy with my 3-month-old son. Recently I became a father, so yeah.
That's a good news.
So yeah,
too much occupied, I think so.
Yeah, too much.
Also, sir, last question, sir. Like, what kind of approximations you took into the consideration? No, like statistical approximation. We have to, when we do this molecular dynamics and all, no, like we have to keep the system on the heat bath, no, like NPT, NVT, and all. So
that has been well defined. That has been quite well defined in Charm GUI, and one of the tutorials from Justin Lemkul. This is the guy who developed this molecular dynamics. And for your knowledge, the GROMACS is developed in Uppsala University. You know that? So I'm fortunate enough to work with one of the founders of GROMACS, who is known as David van der Spoel, here in Biomedicine Centrum. So yeah, you can follow the tutorials from Justin Lemkul. I have shown in my video also, in my YouTube video also. You can follow the tutorial and the approximations for ergodicity. You say as ensemble averages like NPT, NVT. So that you can take. You follow the same tutorial. You don't have to change it. Just follow the same. And it has been maintained at 300 Kelvin. That's the temperature. Typical temperature that we take to equilibrate the system.
Thank you, sir.
Thank you very much, sir.
Thank you, sir. You talk on electronic transport at a nanoscale and nanomaterial complex architecture. It will definitely help our participants on research area. Thank you, sir.
Thank you very much. Pleasure to have you all. Thank you very much.